Today on Incan Insights, we are honored to welcome Dr. Thomas Christian Südhof, Nobel laureate in physiology or medicine and one of the world's foremost neuroscientists. Born in Göttingen, Germany, and now a professor at Stanford University, Dr. Südhof has dedicated his career to unraveling the mysteries of synaptic transmission, the very process that allows our brains to communicate. His groundbreaking work on neurotransmitter release earned him the Nobel Prize in 2013, and his research continues to shape the future of neuroscience and medicine. Beyond the laboratory, Dr. Südhof's journey reflects a life of curiosity, resilience, and a deep commitment to advancing human knowledge. Today, we'll explore his personal story, scientific milestone, and his vision for the next frontier in science. Dr. Südhof, welcome to Ink and Insights with Sumit Sharma Samir.
Thank you.
Sir, growing up in Göttingen, what first sparked your curiosity about science and the brain?
Unfortunately, and maybe disappointingly so, initially I was not interested in science at all. I was more interested in other things, humanities, philosophy, music, all those things that are maybe not that directly useful, though I do think that for society, for humanity, they're actually very useful.
Right. And who were the mentors or influencers that shaped your path toward neuroscience?
My initial mentors were not in science, but were rather in the other realms of scholarship and also actually in music. I only became interested in science when I became a medical student, and I encountered patients, I encountered professors, and I realized that the level of understanding that science has of the human body and of its diseases is extremely limited and continues to be limited today.
Right. And how did your medical training in Germany prepare you for the leap into molecular neuroscience?
I'm afraid it didn't prepare me very well. So there were some intermediate steps. I started working in the lab while I was a medical student and spent a lot of time in the lab already. That time was in biophysics, not in molecular or cellular biology, let alone neuroscience. But it was in a department which had neuroscientists and whose director, with whom I never worked, Victor P. Whitaker, was actually a neuroscientist. I then afterwards moved to Dallas, Texas, in the United States to become a postdoctoral trainee in the laboratory of Joe Goldstein and Mike Brown to work on cholesterol metabolism. That was the place where I really learned how to do molecular and cellular biology, although not neuroscience.
Right.
And these professors were truly my mentors in that they taught me how to do science. I only became a real neuroscientist when I started my own group at UT Southwestern and started my own program, my own research program.
Outside of science, what passions or hobbies keep you grounded?
Well, one thing that really keeps me grounded are the people I interact with, not just family, but primarily actually friends, partners, people I love. The other thing that keeps me grounded is exercise. Just letting everything go and just only focus on physical activity. And finally, reading about what's happening in the world, reading books.
Right. Sir, could you explain in simple terms the essence of your Nobel-winning discovery on synaptic vesicile release?
I can try. When you consider the brain, it contains zillions of nerve cells that collaborate with non-neuronal cells, glia cells, to establish networks. And in these networks, a nerve cell communicates with another nerve cell via synaptic transmission. The synaptic transmission is sending a signal that is a chemical in nature from one cell to the next at a specialized junction, the synapse. At the synapse, the transmission of information is initiated by the secretion of the chemical signal, the neurotransmitter, that is extremely well controlled and regulated. This process of synaptic transmission, which involves the secretion and recognition of a chemical neurotransmitter, also changes the signal, the information that is being transferred. So a synapse forms the ultimate unit in the brain at which information is processed. Our work focused on trying to understand how a synapse is able to send out a chemical signal, the neurotransmitter, in a very brief, incredibly controlled manner. How is it possible that when an electrical signal arrives at the synapse from the presynaptic neuron, that this neuron can then emit the chemical signal within a few hundred microseconds? The chemical signal then binds to postsynaptic receptors on the receiving cell to elicit a postsynaptic response. This process, synaptic transmission, underlies all brain function, and it operates with an amazing speed, precision, but also an amazing plasticity in the sense that it can be changed as a function of either use or as a function of extrinsic signals that act on the pre or postsynaptic nerve cell.
Right. And what was the most surprising moment in your research journey when the data revealed something unexpected?
In science, we operate always by surprises. We look for surprises because we don't know what we're going to discover. It is not possible for us to predict how something is going to work. We think about it, then we test it, and discover that our thinking is often wrong. Many times in my career, I've been amazed how something works, how biology is capable of achieving so many different processes in life. I'm interested not only in synapses, but synapses is what I work on. And at the synapse, for example, I am truly amazed how elegant this molecular machinery operates in very economical ways to be able to convert the electrical signal that travels down the axon to reach the synapse into a chemical secretion of a neurotransmitter. And that is truly for me amazing, that you need only a few proteins to achieve this. And those proteins can bind to each other, interact with each other, respond to signals, and also are responsible in large part for the plasticity of the process.
So how do you balance the precision of molecular biology with the broader questions of human cognition?
Human cognition is obviously what we are all interested in. That and those other things that we humans do, memory, love, consciousness, all those things, our emotions, there's a huge gap, a huge separation that needs to be bridged in order to go from synapses to behavior, broadly speaking, with cognition and all these other things forming part of behavior. The way how we in neuroscience try to approach this is to go from synapses to networks, and from networks to behavior. It turns out that this, although it sounds straightforward, is actually extremely challenging because synapses are very diverse. They differ from each other, so you can't just assume that they're always the same. They're not. They're also plastic. Moreover, networks are very, very complex. There are many different types of networks in the brain. There are local ones. There are ones that are spread across the entire expanse of the brain. They are interdigitated, overlapping, parallel. At this point, although there are some claims to understand the behavior, I'm rather skeptical because I think that, not surprisingly, the complexity of information processing in the brain is vastly more than would allow us to understand, let's say, cognition, even memory, which is a simpler problem than cognition. Just the process of storing some information and then recalling it is, at this point, impossible to approach experimentally, although we are getting closer.
Right. Your work has influenced treatments for neurological disorders as well. Which applications excite you most today?
Certainly not Botox, because that is certainly that is gratifying and rewarding for individuals, but maybe not the most important thing. I believe that our work has the potential, as yet unachieved, to contribute to understanding and eventually, hopefully, also treating what are the most important and pressing needs as regards the brain disorders, brain diseases at present. And these are, on the one hand, neuropsychiatric disorders, autism, schizophrenia; on the other hand, neurodegenerative disorders, such as Alzheimer's disease. I believe that we can contribute, and I hope we are contributing. I hope we have already contributed, because on the one hand, many of the genes that are now discovered to play key roles in schizophrenia and autism are genes that we have discovered and whose function we have studied. On the other hand, synapses are the initial point of vulnerability for neurodegenerative diseases, especially Alzheimer's disease, although nobody at present understands how in these diseases synapses are impaired. There's ample evidence that synapses may be the first, but certainly are among the first parts of the brain that are affected, thereby causing the symptoms that are observed in these dementias. So I think that in our societies, these diseases are an increasing threat to the health span, to the ability of people to live healthily and with a clear mind. And so I'm hoping that we and others can actually use information on synapses, information on these genes that are involved in these diseases to eventually treat them.
धेरै टाइम छैन है बिहे गर्नु छ भने गरिहाल नत्र घरबाट मलाई अन्त्य बिहे गर्दिनु
भर्खर 3 वर्ष भयो एमबीबीएस जोइन गरेको 5 वर्षको कोर्स हो के भो जम्मा छ एक्सक्यूज मी सर मेरो 5 वर्षको कोर्स 3 वर्षमा सकाउन मिल्दैन
गेट आउट
हेलो
के सोच्यो त
के सोच्ने मैले प्रिन्सिपलसँग पनि कुरा गरिसकेँ 3 वर्षमा पनि के हुन्छ 3 वर्षमा
के भयो भाइ ल बस साउजी एक कप चिया कसले भन्यो 3 वर्षमा केही हुँदैन भनेर यो हेर एलआइसी नेपालको धनवृष्टि योजनामा 3 वर्षमा पैसा फिर्ता हुन्छ
ए हो र
जीवन बीमाको साथमा धनवृष्टि 3 वर्षमा एलआइसी नेपालको धनवृष्टि योजना एलआइसी नेपाल
Looking back, which experiment or paper do you consider the turning point in your career?
There are two types of Nobel Prizes. There are those Nobel Prizes that are given for singular discoveries. Often these are Nobel Prizes on techniques, where somebody discovers a new approach for addressing a problem. A typical example is the patch clamp Nobel Prize to Neha and Sackman, or the antibody, the monoclonal antibody Nobel Prize a decade earlier. Then there are also the Nobel Prizes that are given for addressing a biological problem, a biological question. These Nobel Prizes usually do not recognize a singular paper or even a few papers. They basically recognize a body of work because understanding a biological process involves many incremental steps that are published incrementally. So there is no paper from my own work that I consider to be the most important paper. Instead, there are several that I consider key increments in the overall discovery. And these involve, for example, the discovery of synaptotagmin, the discovery of MUNC18, the discovery of RIM, the finding what these proteins actually do, how they interact, things like this. But it was really 1,000 steps that led to the overall discovery, not just one particular observation.
Dr. Südhof, how did receiving the Nobel Prize change your life, both professionally and personally?
There were good things and bad things. The good things, obviously, is that I can request to fly business class, which I appreciate. And especially as I get older, and that I have an opportunity, at least sometimes, to meet a lot of interesting people and to try to advocate for causes that I would like to support. The bad thing is that, especially if you're a Nobel laureate who continues to do science, you attract the attention and sometimes even the hatred of people who don't like you for a number of reasons. And so I have felt under a microscope scrutinized to an extent that I didn't think was possible. And that certainly would not have happened if I didn't have the Nobel Prize.
So do you feel the Nobel Prize has shifted expectations of your work?
It's not the expectations of the work. It's just like, I guess if I had simply become an elder statesperson of science, like many Nobel laureates do, people would have been very happy and would have left me alone, and I could have traveled the world. That is not what I wanted to do, and that's not what I do. And so, as a result, I become basically attacked and scrutinized on a regular basis, which is, I guess, the dark side of science in general these days. As you may know, in many respects, science has entered a very difficult time, not just in the United States, but certainly in the United States. And it is changed because I think people are just, many people, maybe justifiably, are disappointed at the current state of life, of things in the world.
I can imagine that. What advice would you give to young scientists who aspire to make discoveries of global impact?
I'm not sure if I'm qualified to give advice because I am primarily a practicing scientist and I try to train people. The advice I give to the people I train when they leave the lab and move on to their own career, new careers, is that they should always try to focus on what I call the content of science and try to distance themselves from the peripheral stuff, from the formal stuff, from all the outside things. Of course, you can't dissociate yourself completely because everybody is part of society. We are part of society. We need to be within society and we work for society. Our work is meant to benefit society. But in doing so, we need to focus on the actual discoveries that we are trying to work, on the process of science, on discovering things, on observing things. We should not focus on things like recognitions and papers and, all these outside things that can basically be very disappointing or sometimes also very nice, but in general, this is only a distraction. What is really important is for a young scientist as much as for an older scientist like myself to keep our interest, faith, focus, concentration, whatever you want to call it, passion, whatever, on the actual process we are trying to understand, on the biology, on the content of what we do, and let the societal and recognition and other things, promotions, papers, yeah, that's important. We can't ignore it. But it is secondary to the actual discovery process.
Absolutely. And where do you see neuroscience heading in the next 20 years?
That is a great question because we are experiencing, in many ways, a technical revolution. In general, in neuroscience as well as in the wider world, we, I believe, have no idea where AI will lead us, how good it can be, or possibly not so good. Whether there are even dangers in AI that will destroy science, that will destroy societies. So AI will be clearly a huge component of everything we do in future. But it's not the only one. There's also other technical innovations that have changed how we do science, including neuroscience. There are innovations such as cryo-EM, which allow you to get structures of molecules much more quickly and bigger molecules, bigger assemblies. There's things like transcriptomics, there's CRISPR, there's so many other new technologies. We are just starting to use these. And the challenge is to integrate them and to make use of them without becoming tethered to one particular approach, without becoming sort of limited in our vision, in our understanding, because these different approaches are so different. Thus, we really need to try to get an integrated overview. We need to try to achieve a synergy of these approaches, including AI, but not only AI.
Right. Dr. Südhof, what ethical challenges do you foresee as neuroscience uncovers deeper insights into human behavior?
So there's several levels of challenges. Of course, the general challenge of AI is not in neuroscience, but rather in its ability to track everything, to remember everything, to retrieve everything. That is not a neuroscience challenge. That is a societal challenge, which I believe most governments are currently ignoring, and in fact, purposely often. So I'm a bit worried about that. In neuroscience, ethically, the challenges are similar to those as in other parts of biomedical research. Who will benefit? Can we develop ethically treatments for diseases that are going to be available for people who are poor, for countries that are poor? I'm not so sure that brain-machine interfaces are going to be that useful for a lot of people because they will be insanely expensive. Right. I think that ethically we need to focus on innovations and advances on progress that will actually help large populations in future. And so ethically in science in general, we need to make sure, first of all, that science will always be open, that knowledge will be for humanity and not just for little countries everywhere. And second, that open society and open knowledge, open scholarship, that leads to insights which we can use to treat diseases that are common diseases in a manner that is accessible for as many people as possible.
Absolutely. My last questions for today's program, Dr. Südhof, when you reflect on your career, what legacy do you hope to leave for future generation of scientists?
I'm hoping for two legacies. One is, I'm hoping that the people who have worked with me and who have gone on to their own independent careers, that they will be able to take something away from having worked with me that will enable them to make major discoveries on their own and to advance knowledge for everybody. My other legacy that I'm hoping for is that what we have discovered and are continuing to discover will form a fundament on which others can build to achieve what I said earlier, which is eventually to achieve an understanding of how the brain works and how its functions become impaired and diseased so we can change the diseases and we can treat people. Those are the two legacies that I'm hoping for. Maybe that's too ambitious, but that's what I would love to achieve.
That's achievable. I wish you good luck for that, Dr. Südhof. And, one key takeaway, which kind of really touched my heart today during the conversation is that we should not be, focusing more on societal recognition, but much more into the content of whatever we do. Thank you so much for your time, Dr. Südhof. I had been chasing you for almost last 1 year now, and I'm deeply humbled for your time and participating in my platform. Thank you so much.
Thank you.